Good afternoon, everybody. Thanks, Lindsay, for the intro. Wanted to go over where we are at Homology Medicines and what we're doing. In this brief period of time, we are a public company, so you can check our SEC filings on our website. I recognize I'm in the lead-off position here, so I'm going to have to, keep the pace. As was just mentioned in the previous panel, we're at a really amazing time right now. We can think about potentially curing patients. This is certainly what drives all of you in the audience, working to bring these transformational therapies and treatments and potential cures to our patients. Certainly what drives the culture at Homology. We feel like we have a differentiated platform of 15 novel AAV. We call them HSCs because they were isolated from hematopoietic stem cells that have the ability to drive gene transfer, the delivery of episomes, and or gene editing, or the precise insertion of genes into the genome via homologous recombination. On the right-hand side, you can see we started the company late 2015. Management team came on board early 2016. It's taken us 3 short years to drive all the way through our lead program into the clinic, and we're conducting a phase 1/2 trial right now for gene therapy for adult patients with PKU. In the lower left-hand corner, we have a team that has been involved in rare disease drug development for quite a while. These are some of the examples of what we've brought forward. None of these are curative, and that's why we're so excited to be able to talk about the homology technology today. And we have broad IP on composition of matter, methods of use for these vectors. So really, this is the Bimodal Platform on the left-hand side. It's your basic gene transfer cassette with ITRs flanking a promoter and the gene of interest. And this is meant to represent our AAV HSCs. This will deliver episomes through very good transduction capability into the nucleus and drive expression off of that piece of DNA. On the right-hand side, what you see is the gene editing construct. So you can imagine within the same vector, the same AAV HSC, and this happens to be AAV HSC 15, you can have a slightly different single-stranded piece of DNA that has ITRs flanking homology arms, which direct that gene of interest precisely into the genome where you want it to be, and with a very, very low error rate, which is what this DNA repair mechanism is known for, very tight precision. And the way we think about this on the left-hand side, we have an advantage in gene therapy because we have 15 of these naturally occurring AAVs that have very good but slightly different tissue tropisms. So you can select one if you want to go after the brain, you select another one if you want to go after the liver. And so we have that ability on the gene therapy side. On the right-hand side, we separate ourselves from the nuclease-based approaches where you're making a cut with a nuclease in terms of being able to cut a gene. Here, what we're focused on are autosomal recessive disorders where both alleles are affected, and you're going to need the insertion, the precise insertion of a gene in order to effect a cure. This is a look at our pipeline. We have gene therapy programs. Those are the first 2. That's gene therapy for adult patients with PKU, HMI-102, which is currently in a phase 1/2 trial, and due to report out the first cohort of patients in terms of safety and efficacy at the end of the year. The second program we have in the queue is a gene therapy program for a PNS disorder called metachromatic leukodystrophy, or MLD. This is a terrible, pernicious disease. Usually these patients die in the first decade of their lives. And here we're going after replacing the enzyme that's missing in this disease, which is aryl sulfatase A. And then below that, you see our lead gene editing program, which is a editing construct, HMI-103, for the pediatric population of PKU. As you know, this is a quite common, rare genetic disorder, and to be able to go early in treatment and make sure that you have that insertion and that is durable is what you want for a lifelong cure. And we have some discovery programs, including an in vivo approach to hemoglobinopathies, as well as our partnership with Novartis, which focuses on eye diseases. And this is utilizing the gene editing component of our platform. So really, let's talk about PKU. This is a disease where you're unable to make PAH, phenylalanine hydroxylase, and that enzyme, as you can see on the left-hand side in green, is responsible for converting phenylalanine to tyrosine. And tyrosine is an important precursor of the neurotransmitters and of melanin. When you go after a PAL approach, this is phenylalanine lyase, and so this is essentially Palynziq and other approaches, you're delivering a non-human enzyme which is going to degrade phenylalanine, not to tyrosine, but to two other metabolites, trans-cinnamic acid as well as ammonia. So here what we're trying to do is reconstitute the naturally occurring enzyme that's missing in these patients. And this is the mouse model, the U2 mouse model, which has been used for many, many years to look at different therapies for PKU. This mouse is missing the PAH enzyme, and you can see untreated or placebo-treated mice on the left-hand side. That's that top bar run at about 1,500 micromole phenylalanine. That's really high, and phenylalanine is toxic to the CNS. After one administration of HMI 102, this is a gene transfer construct, you drop that all the way down to normal levels for the lifespan of this particular mouse model. On the right-hand side, this is a biochemical analysis that a lot of doctors will run, is you're looking at the phenylalanine to tyrosine ratio, and as you can see, one treatment of HMI 102 normalizes this down to normal levels of which you would expect in normal mice. So this is a dramatic improvement in terms of being able to reduce Phe in a consistent way. Here what we did is to really kind of highlight the transduction capabilities of the AAV HSCs, and we compared it to AAV5. When you package AAV5 and package AAV HSC15 with the PAH construct, the low dose here on the left-hand side is our starting clinical dose. It's in the E13 VIGs per kg range. You can see we are able to drop the AAV HSC15 all the way down to normal. AAV5 does not have that effect at all. It's very close to placebo. On the right-hand side, on the high dose, this is our second cohort dose in the clinical trial. You can see now with a higher dose, AAV5 starts to have some activity, but not comparable to AAV HSC15. This is really meant to highlight the tropism of these vectors for the liver compared to other AAVs. And here we're showing normalization of phenylalanine in the brain. So on the left-hand side, it's normal mouse levels. In the ENU2, that naturally goes up. And with a single administration of HMI 102, that comes back down to normal. And so that's normalization of brain Phe, which is very important. What I just showed you before was serum Phe. In the middle panel, what you see is an increase in brain 5-HIAA, which is a metabolite of serotonin. You can see in the yellow, that's what normal mice look like. You want serotonin levels to be high. In the ENU2 knockout mouse, those animals have very low levels, but you normalize that with that single injection of HMI 102. And perhaps the most striking is the biochemical conversion in the upper part of the right-hand panel. These are your ENU2 mice. They're typically blonde. And after a single treatment, after a couple of weeks, these mice begin to turn black, which is what is expected if you're making melanin. So it's a very good visual indicator that you're doing what you're supposed to do. What we also have done is to compare the tropism of these vectors in male and female mice and the number of vector genomes per cell, if you will. On the left-hand side, again, the low dose, our starting clinical dose. You can see the levels there in male and female mice, very comparable to non-human primate levels in the liver. And that's an important step that we took to try to see what the dose translation is going to be in a higher primate. And again, on the right-hand side, you see a similar result with our mid-clinical trial dose. So that's comforting that we're seeing that translation from mouse to non-human primates. This is the FIXX trial. So this is a high level, it's an open label, randomized concurrently controlled dose escalation study in adult patients with PKU. Primary objective is safety, of course. Secondary, the efficacy endpoint here is reduction of Phe, the consistent reduction of Phe, below 360 micromole. Below 360 micromole is important because that's what the American College of Medical Genetics would recommend for patients to come off their restrictive protein-free diet. It's a single administration IV of HMI 102. Patients will be between 18 and 55 years old. We have the ability to do 3 dose cohorts as we escalate and the ability to add an additional cohort. And this is also has the ability to be converted to an expansion trial. So it has a part A, which is the dose escalation, but if we reach a dose that we feel is effective, we're able to add more patients to that dose and concurrent controls, with the ability to take those controls after a certain period of time, in this case 24 weeks, and treat them and roll them over to the treatment arm. So those 9 patients would be considered as part of a registrational trial. So we have flexibility here to move quickly to the right dose. That's gene transfer. Moving now to the gene editing platform. This is essentially the same mouse model I just showed you, but with an editing construct, which is HMI 103. And that, again, drops phenylalanine down below 360 consistently. And you can see the normalization of the Phe-tire ratio as well. Here we're showing mRNA levels produced by this editing construct. And this was a difficult thing to do for the research team to do. Under Albert Seymour, we had to show in a human liver tissue that a human clinical development candidate can, in fact, precisely insert the PAH gene where it's supposed to go and then drive mRNA expression off that. On the right-hand side, what you see is the murine construct, and you can see the levels of mRNA reach around 40% compared to a housekeeping gene. That 40% level is about normal level, so that will get you reconstitution and dropping a Phe to where we want it. On the left-hand side, we used a humanized liver model. So this is a immunosuppressed mouse with a human liver, and we used that to show that the human construct that we will bring forward into clinical trials can precisely insert and generate the mRNA at the same level required to reduce Phe. So the comparability here is important, and the ability to show mRNA expression with the human construct in human liver tissue is also very important. Wanted to highlight the biodistribution properties of these vectors. We've done a lot of non-human primate studies looking at these different vectors, all 15 in fact, and we have publication going out shortly, if not already, on this. But we have very good tropism to the liver, very good ability to cross the blood-brain barrier, which is why we're going after metachromatic leukodystrophy, as you can see here in the lower left panel. Gets to the lung, skeletal muscle, cardiac muscle, which is not there, and to the back of the eye after a single IV administration of these vectors. And so MLD, I gave you a little bit of the struggle here with this disease. It's a rapidly progressing disease where you are missing an enzyme that degrades sulfatide, and that sulfatide is toxic to neurons. And so the key is to be able to reach a level of aryl sulfatase A, not only in the brain, but peripherally, in order to have the benefit that you would like to see, to have the chance to cure this disease. What you can see on the left-hand side is with increased dose in the brain, you can see increased expression of aryl sulfatase A, or ARSA. In the lower left-hand side, you can see the biodistribution throughout the mouse brain in the ARSA knockout. So those dark staining punctate marks are GFP expressing, that's being expressed by our AAV HSC. And then on the right-hand side, you can see reconstitution in the animal model of aryl sulfatase A levels that are comparable to wild type. Last couple of slides, we have built our own internal manufacturing capability. It's a 25,000 square foot facility. This is already producing GMP material for our clinical trial right now. This is a core capability of Homology. We invested very heavily in this early because we know that manufacturing is usually the timeline killer in gene therapy drug development. And we have an outstanding team led by Tim Kelly and many of the folks that worked with him at Shire in really developing a single-use commercial scale. We're already at 500 liters plus GMP material for the trial and for our other programs and the rest of the pipeline. So it's a big year for us. In 2019, again, we'll be reporting out phase 1/2 data, at least in the first cohort, for the adult PKU gene therapy trial. We are in IND enabling for the pediatric PKU program with HMI 103, IND enabling for the MLD gene therapy construct, and again, very rapid progression of our manufacturing capability, and we'll continue to advance the rest of our pipeline in CNS, ophthalmology, and hemoglobinopathies. Thanks for your attention, and it's nice to see so many colleagues in the audience, and enjoy the rest of the meeting. Thank you.